Hi everyone, welcome to my channel Pure Biology, and today I come with another video. Obviously, I will try to explain about the action of the mode of action of the pertussis toxin in case of whooping cough. Okay, this is very brief topic, and I just explain how this action is created due to the cell signaling process. Okay, and before going to this topic, I want to say thank you to all of you and who have already subscribed my channel. And it's my humble request to the newcomers, please you just share and subscribe this channel if this is helpful to you. Okay, and your support is very needed for me. Okay, let's continue. Mode of action of pertussis toxin. So here the pertussis toxin is an exotoxin. Okay, this is an exotoxin and that causes the pertussis and it's a whooping cough. Okay, and this foreign particle is the Bordetella pertussis. Okay, the toxins that is secreted from the Bordetella pertussis, and it is very harmful to us because it creates whooping cough. So here first we have to understand that actually what is the function of the ciliated epithelial cells. Okay, so first these ciliated epithelial cells are present where? These are present within the inner surface of the respiratory tract. Okay, so what's the function of this? In the respiratory tract there the ciliated epithelial cells are present. These epithelial cells actually functions as it helps to sweep away the mucus. Okay, these ciliated epithelial cells actually try to sweep away mucus from the respiratory tract where it just coagulated. Okay, so its function is that any mucus or no mucus, you can say, that is stored within a particular area within the respiratory tract. So that's why these ciliated epithelial cells, they always try to sweep away this mucus along the tract. So here due to the infection of the Bordetella, its function is what? They actually destroys the ciliated epithelial cells. When any respiratory tract that is infected by the Bordetella, then the ciliated epithelial cells are all destroyed within the tract. Okay, and so what happened? This ciliary action is stopped. Okay, and for this ciliary action, or you can say that to sweep away the mucus, we need to vigorous cough or vigorous coughing. Vigorous coughing is very needed for us to clear the tract or to function like that ciliated cells. Okay. So here actually happening that thing, the pertussis toxin catalyzes the ADP ribosylation of the cysteine residue of alpha subunit of GI. Clear? GI means GI protein. So what's the function of this toxin? This toxin catalyzes the ADP ribosylation of cysteine residue of alpha subunit of GI protein. Generally, GI protein is an inhibitory protein. Okay, GI protein is an inhibitory protein. So why it is called the inhibitory protein? Because in activated state, GI alpha binds to GTP and dissociates from the beta gamma complex, and the released GI alpha inhibits the adenylate cyclase. Clear? Adenylate cyclase. So if this is occurring in normal situation, so no coagulation of the mucus is possible within the respiratory tract. Okay, because adenylate cyclase level is very low. But what happens when the infection is occurring within the respiratory tract? Then the ADP ribosylation of GI alpha that prevents, okay, that means ADP ribosylation of GI alpha that is stopped, and the GI complex from the interacting with the receptors. So the complex remain bound to the GDP. So what is saying that the ADP ribosylation of GI alpha that prevents the GI complex from interacting with receptors. That means no receptor can bound with this GI complex. So dissociation of the GI alpha from the beta gamma is not possible, and as a result, what happening? This remain bound to GDP. The complex remain bound to GDP and unable to inhibit the adenylate cyclase. So if the adenylate cyclase level is not controlled, the adenylate cyclase level rises, and what happening in that situation? The coagulation of the mucus is occurring within the respiratory tract. So in the normal situation, what is happening? Generally GI alpha that released, this GI alpha released or dissociates from the beta gamma complex, and it inhibits the adenylate cyclase level, or it controls the adenylate cyclase level. And the ADP ribosylation, what happening in this ADP ribosylation of the GI alpha? This prevents the GI complex from interacting with the receptors, so dissociation is not occurring and the complex remain bound with the GDP. And what is happening in that situation? That inhibit the adenylate cyclase. That means that unable to inhibit adenylate cyclase. That means they cannot control the level of the adenylate cyclase. So this situation is seen in the whooping cough or the infections caused by the Bordetella within the respiratory tract. So this is one kind of signaling process, or you can say the situation is created just like the signaling process. Here the G protein has important role for this creation. Okay, so this is a very short topic about the pertussis toxin, or how the signaling process is occurring here. Okay, so in the next video I will try to explain another topic. So please continue with my channel.